Hello everyone, welcome back again to the series of solved previous year's ICAR PG examination Plant Science, where we will discuss the questions on genetics and plant breeding. Today we will discuss the paper of 2021, so let's get started. Question number 1. If a woman carrier for color blindness marries to a colorblind man and produce several children, the expected phenotype of the offspring would be. So here it is given, the female is the carrier and the male is colorblind. The offsprings produced would be these, and we can see here the woman is the colorblind or the carrier, and the males, they can be normal also or colorblind. So option 4 will be the correct, because all females will be carriers of colorblindness or they would be colorblind. Question number 2. In Q banding experiments, the light fluorescent band indicates that the chromosomal region is rich in base pairs. They are rich in G and C base pairs. This Q banding, it is used to identify the individual chromosomes and the structural abnormalities in banding patterns. It uses the chemical quinacridine. Question number 3. Horn development in sheep is a suitable example of which of the following events? So it is a sex-influenced trait. It is a trait which is influenced by a particular sex, that is, horn is dominant in males whereas it is recessive in the female sheep. In case of sex-limited traits, these are the genes located on autosomes which are expressed only in one sex. For example, barred color is present only in the roosters. The sex-linked traits, they are the traits which are determined by the genes located on one of the sex chromosome. For example, hemophilia. Then we have Hollandric traits. These are the traits that are passed on from father to son. For example, the webbed feet. Question number 4. The genotype of a plant is this. The genes capital A and capital B are linked in coupling phase with a map distance of 10 centimorgan. The gene capital D is present in a different chromosome. Under such situation, the frequency of capital A, small b and capital D gametes would be. So we are given that the chromosomes are in coupling phase and the distance between them is 10 centimorgan. The recombinants formed, they are 10 centimorgan total. So recombination will be divided as 5% each, whereas the parents, they have 90%, so 45% each. We are talking about capital A small b and D genome, that is this. For a different chromosome, we use 1/2 frequency. So 5 will be divided by 100 into 1/2, which gives the answer 1/40. So the correct answer is option 4, 1/40. Question number 5. A tall green gram plant was crossed with a dwarf plant. All the F1 plants were of intermediate height. The F2 plants produced different phenotypic classes in the ratio of 1:4:6:4:1. How many genes are involved in controlling the plant height? So here 2 genes are controlling the character, that is option number 2, because only the part of trait is expressed by a single dominant allele. The phenotype of offsprings depends on the number of dominant alleles, so 2 genes are there which represent the following ratio. Question number 6. In a population of 100 individuals, which is in Hardy Weinberg equilibrium, the number of individuals of different phenotypic classes are as follows: 64, 32 and 4. The frequency of capital A and small a in this population would be. So from 32 AA we can find out that both the alleles have 16 frequency. For capital A it is 64 plus 16, which is 80, and for small a it is 4 plus 16, that is 20. For calculating the frequency of capital A, it is the number of A upon the total individuals, that is 80 upon 100, which gives the value 0.8, and for small a it is 20 upon 100, which gives 0.20. So the correct answer is option 3, 0.80 and 0.20. Question number 7. The most common secondary structure of protein molecule is, it is the alpha structure. The alpha helix is the most common secondary structure motifs formed in proteins and the polynucleotides. Colchicine is primarily obtained from which parts of the plant? It is obtained from the corm and seeds. Colchicine develops polyploid plants and is called a mitotic poison which inhibits the formation of metaphase. Question number 9. On average, how many fragments would a restriction enzyme which recognize a specific 5-base sequence in the DNA would produce a double-stranded bacteriophage genome of size 525 base pairs? So we know that A is equal to T and is equal to G, which is equal to C. It is 1 by 4. So we are given 5 base sequences. So it will be 1 by 4 raised to the power 5. That is 1 by 1024 into the total base pairs, that is 525, which gives about 5.1, that is approximately there are 5 segments. Question number 10. The concept of chromosomal non-disjunction was given by C.B. Bridges. It is when the separation fails to occur during anaphase and both the sister chromatids they are pulled together at one pole of the cell. Question number 11. Given below are two statements. One is assertion and the other is reason. So assertion says that in reciprocal recurrent selection, the two populations so developed can be used as a synthetic variety. Reason is in reciprocal recurrent selection, the GCA of both the population is improved simultaneously. So both the varieties, they improve each other characteristics and they are also improved simultaneously. So both the assertion and reasoning, they are true, and R is the correct explanation of the assertion. Question number 12. Given below are two statements. One is assertion and the other is reason. Assertion says that Brassica juncea is an allotetraploid having capital A and capital B genome. Reason says that Brassica juncea is produced when Brassica nigra is crossed with Brassica campestris. So Brassica juncea, it has N is equal to 18 and it is allotetraploid because Brassica nigra, it is BB genome which is N is equal to 8 and Brassica campestris, it has genome AA and N is equal to 10. So assertion and reason both are true and reason is the correct explanation of A. Question number 13. Cis-trans or complementation test results may be ambiguous if ambiguous means to be doubtful. So when the mutations are dominant, the complementation can be doubtful. When there is intragenic complementation, there is polar mutation, or when there is gene interaction. C is actually what is complementation test. So it is the mutations belong to different genes with different phenotypes. So the correct answer would be A, B and D, E. That is option number 3. Question number 14. The events happening during RNA processing are, so first, the capping at the 5' end occurs. After that, splicing of introns and joining of the exons occur. Later, addition of poly A tail and 3' end occur. So the correct answer would be A, C and E. That is option number 2. Question number 15. Chronological steps in the development of genotype and release of a crop variety are. So first of all, there will be genotype development. After that, preliminary yield trial will occur. Following, we have the multi-location trial. Then identification of the variety. After that, the notification of the variety. So the correct answer is option 2, B, A, C, E and D. Question number 16. Chronological steps in the development of hybrid are. First, we will develop the inbreds. After that, morphological evaluation will be done of the inbreds. Then we have the testing for combining ability, followed by the evaluation of the hybrid with check variety. Later, the seed multiplication will be done. So the correct answer would be option number 3, that is A, D, C, B and E. Question number 17. Given below are two statements. The light-dependent reactions occur in the stroma in the presence of light. The light-independent reactions occur in the thylakoid in the absence of light. So both the statements, they are wrong because light-dependent reactions, they occur in thylakoids, whereas the light-independent, they occur in stroma. So both the statements, they are incorrect. Question number 18. Given below are two statements. Statement 1. Matthias Kleeden and Theodor Swan proposed the first two components of the cell theory. Statement 2. Rudolf Virchow proposed the third component of the cell theory. So both the statements are correct. Question number 19. Given below are the two statements. Pre-1940, genes were considered to be like beads on string and not divisible. In 1940, C.P. Oliver proved that the genes are divisible. So both the statements are correct, and it says, earlier it was said that the crossing over does not occur within a gene, it occurs between separate genes, but later it was proved that the genes are divisible. Question number 20. Given below are two statements. Plastids are sites of purine synthesis. Cytosol is the site of pyrimidine synthesis. So both the statements are correct. Question number 21. Single cross hybrids can be used for a variety of purposes such as, so they can be used for the development of three-way cross hybrid. They can be used for the development of double cross hybrid. Also, they can be used for the prediction of double cross hybrid. So the correct answer is option number 3, that is A, C and D only. Single cross, we know that when inbred A will be crossed to inbred B, we have a single cross. Question number 22. For estimation of mid-parent heterosis, the genotypes included in the trials are, we know for the estimation of heterosis, we need a female parent which is to be crossed to a male parent so that we get F1, which is then checked the heterosis. So we require F1 hybrids, female parents and male parents. The correct answer would be option number 2, A, B and C only. Question number 23. Heterosis cannot be fixed if it is obtained through. So if heterosis is obtained through the development of F1 hybrid or through the development of synthetic variety, the heterosis is not fixed. The correct answer is option number D, that is C and D only, because in these cases the heterosis is exploited. Heterosis is fixed only by the use of apomixis, lethal system or vegetative propagation. Question number 24. Frequency of recombination between two genes is affected by, it is affected by A synapsis and D synapsis. It is affected by the location of the gene with respect to the centromere, the distance between two genes and the translocation of the genes. So the correct answer would be option number 3, B, C, D and E only. Asynapsis is the situation, that is the failure of the homologous chromosomes to pair or synapse during the first meiotic division. Whereas desynapsis, it is when the homologous pair, they normally pair at the prophase but later fail to maintain their association. Question number 25. Match the human disease or traits with the onset of expression. So L-captanuria, it is expressed about immediately after birth. The blood groups, they are determined before birth. Baldness, it comes about 20 to 25 years of birth. Rickets, it appears about 1 year after birth. So the correct answer would be A3, B1, C4 and D2. So option number 2. Question number 26. Match the theory or principle with the names associated with it. Central dogma of molecular biology is related to Francis Crick. Chromosomal theory of inheritance was given by Sutton and Boveri. DNA double helical structure was given by Watson and Crick. One gene, one enzyme hypothesis, it was given by Beadle and Tatum. So the correct answer would be A3, B4, C1 and D2, that is option number 1. Question number 27. Match the sex index with the sexual phenotype. When 1 is there, that is associated to female, 0.5 is associated with male, 0.67 tells us that it is intersex, 0.33 tells us that it is a super male. So the correct combination is A4, B1, C2 and D3, that is option number 1. Question number 28. Match the types of hybrid with the parental combinations of the crosses. A three-way cross hybrid is given as inbred A into inbred B, and completely the F1 will be multiplied by inbred C. Modified single cross hybrid, it is given by inbred A into A dash. The progeny is then crossed with inbred B. Double cross hybrid, it is given by inbred A into inbred B, that is crossed to the open pollinated variety. A top cross hybrid is given by inbred A into an open pollinated variety. So the correct answer would be A3, B4, C1 and D2, that is option number 3. Question number 29. Match the name of the scientist to the significant contribution in the major fields. So Hugo de Veres, he is related to mutation. Walter Fleming, he gave about mitosis and coined the term mitosis. Stern, he tells us about the cytological basis of crossing over in Drosophila. Michelson and Stahl, they gave the semi-conservative model of DNA replication. So the correct answer would be A3, B1, C4 and D2, that is option number 3. Question number 30. The acronym ITPGRFA stands for, it stands for International Treaty on Plant Genetic Resources for Food and Agriculture, that is option number 2. This promotes and facilitates the conservation and sustainable use of plant genetic resources for food and agriculture. Question number 31. In sea urchin DNA, which is double-stranded, 10% of the bases are shown to be cytosine. Based on this observation, the following were recorded. Guanine is 10%, adenine 40%, thymine 20%, purines 50%. Purines and pyrimidines are equal. We know that purines A and T, pyrimidines G and C, so they are equal. That means guanine are 10%. This is right. Adenine plus thymine is equal to guanine plus cytosine, so they must be 40 to be equal to the pyrimidines. That is, purines are 50% and purines and pyrimidines are equal, and adenine is 40%. So the correct answer is option number 3, A, B, D and E only. Question number 32. Most commonly used enzymes for DNA manipulation include the following. Nucleases, they cut the DNA, so they are molecular scissors. Ligases, they work as joining or binding enzymes. Polymerase, they make the complementary strands. Kinases, they perform in pyruvate synthesis. Helicases are used to break the hydrogen bonds. So the first 3 enzymes are used in DNA manipulation. The correct answer would be option number 3, A, B, C only. Question number 33. Which of the following is a form of ex-situ conservation? Ex-situ, when the repository is outside the plant origin. So they are gene banks, tissue culture repositories and botanical gardens. So that the correct answer is option number 1, A, B and E only. Question number 34. Match the list 1 with list 2. Discovery of human blood group. It was done by Landsteiner. Double helical model of DNA was given by Crick, Watson and Wilkins. Genetic improvement of Mexican wheat, that was done by Borlaug. Mobile genetic elements in maize, also known as the transposable elements, was given by Barbara McClintock. So the correct answer is A3, B2, C4 and D1, that is option number 4. Question number 35. Match the list 1 with list 2. Protogyny, it means when the pistil matures before the stamens. Androgyny is related when the stamen matures before pistils. Dichogamy, it occurs when the differential maturation of male and female flowers occurs. Cleistogamy is when the flower does not open. So the correct answers, they are A3, B2, C1 and D4. That is option number 2. Question number 36. Match list 1 with list 2. Rice is the first genetic modified organism for vitamin A. Tobacco is the first genetic modified organism. Tomato, it is the genetic modified organism for shelf life. That is the famous flavor saver. Cotton, it is the GMO hybrid. So the correct answers, they are A3, B4, C2 and D1. That is option number 1. Question number 37. Given below are two statements. One is labeled as assertion and the other is labeled as reason. Assertion says that in Watson and Crick model of DNA, each turn of the helix is the length of 10 base pairs. Reason is each complete turn of the double-stranded DNA helix is 3.4 angstrom long. The assertion is correct and the reason is wrong because the turn of the helix is 10 base pairs, whereas the complete turn, it goes around 34 angstrom and the distance between two bases is 3.4 angstrom. So the correct answer would be that assertion is true and the reason is false. Question number 38. Light pathway of a typical microscope consists of several items. Viz. So the correct answers, the combination is the light source goes to the mirror, after that diaphragm to the condenser lens, then it goes to the stage and the specimen, then the light moves to objective to the specimen, after that the ocular lens to the eyepiece or the eye camera. So the correct answer would be option number 3. Light path is from light source to mirror, to field diaphragm to condenser, stage to specimen, objectives to specimen, and the ocular to the eye or camera. Question number 39. Which one of the following cell organelle does not contain DNA? So the correct answer is option number 1, that is lysosome. Which of the following statements are true while comparing prokaryotes and eukaryotes? Prokaryotes have 70 S ribosomes, whereas eukaryotes, they have 80 S ribosomes. So this is correct. Nuclear material is linear in both the cases. No, because the prokaryotes, they have circular genetic material. Meiosis is the characteristic of the eukaryotes, so it is correct. Genetic exchange occurs in both prokaryotes and eukaryotes. This is also correct. Nuclear membrane is present in both the prokaryotes and eukaryotes. This is wrong because nuclear membrane is absent in prokaryotes. So the correct answer is option number 1, A, C and D only. Question number 41. Given below are two statements. Operon models of gene regulation are applicable to prokaryotes only. Eukaryotic DNA contain intervening pieces of DNA called introns. The first statement is wrong because the operon models of gene regulation, they are also applicable to eukaryotes. Whereas the second statement is correct. These pieces, they are called introns. So the correct answer would be option number 4. Statement 1 is incorrect, but statement 2 is correct. Question number 42. Which of the following are not chain initiation codons? So they are option A, UAA, option B, UAG, and option C, UGA. These are known as stop codons. The correct answer would be option number 3, A, B, and C only. So that was it. Thank you.